What makes us, us? Our genes, of course.
We all have a unique genetic code made of different genes coding for proteins. Having millions of genes, there is room for mutations to occur. Most mutations are insignificant, but if a mutation harms the body, the consequences can be very severe.
Is there a way we can fix these mutations? What an amazing question! A new gene editing technology could be just the thing we need. The CRISPR-Cas9 system is a new gene editing technology that will likely revolutionize the way we approach genetic disease and diagnostic testing. CRISPR stands for Clustered Regularly Spaced, short, palindromic, repeat. That's a mouthful, so let's just stick to calling it CRISPR for short.
So, what makes it so useful besides the fact that it's an amazing tongue twister?
Wow, another amazing question. With CRISPR, scientists can edit specific genes by inactivating them, changing their DNA sequence, and changing how much the gene is used by the cell. Compared to other gene editing technologies, CRISPR is fast, cost-effective, and target-specific. For this reason, it can potentially be used in humans directly, not just cells in a petri dish. CRISPR also has the potential to revolutionize the biomedical industry. From the rapid generation of GMOs to curing and diagnosing genetic disease, and eventually generating pigs and cows that can donate organs to humans. So, now that you're hooked, I'll tell you how it works. CRISPR takes advantage of a cell's normal DNA repair mechanisms, which kick in when damage to the DNA is detected.
Scientists first determine the gene sequence they want to target. For example, a single nucleotide mutation in the hemoglobin gene causes sickle cell. If a scientist wanted to change the mutated gene back to normal, they would create a small guide RNA that's complementary to the gene sequence, allowing the RNA to find and bind to the sequence in the genome, but only that sequence. The guide RNA also has a sequence that allows it to interact with a DNA cutting protein called the Cas9 endonuclease, which forms the Cas9 complex. When the Cas9 complex is introduced into cells, the guide RNA directs it to the part of the hemoglobin gene to be fixed. The cutting protein cuts both strands of the DNA, and the cell's DNA repair machinery comes in to fix the damage.
All right, class. I'll leave you with a relevant example of CRISPR's use today. Scientists have been using CRISPR to develop an extremely fast way to identify the COVID-19 virus. CRISPR is already in development to identify individuals infected with other viruses, and there is hope that CRISPR can be used to selectively disable viruses, cutting the long wait time for vaccine development. Overall, CRISPR is extremely useful in different ways, and now that we've talked about this, don't get too excited and change your genes wildly or you'll end up like this guy.